Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year 1907 witnessed the publication of a seminal work that fundamentally altered the landscape of infectious disease treatment, the experimental chemotherapy of Spirilloses, authored by Paul Ehrlich and Sahachiro Hata. This treatise, documenting their breakthrough discovery of salvarsan as an effective cure for syphilis, represents a cornerstone in the development of chemotherapy. This essay undertakes a detailed, section-by-section examination of the work, critically assessing its claims, methodologies and underlying logic within the context of its historical significance. Section 1, Introduction, and the concept of chemotherapy. Ehrlich and Hata's monograph opens by establishing the theoretical framework for their experimental endeavors, the concept of chemotherapy. They outline their vision of creating chemical compounds capable of selectively targeting and destroying disease-causing microorganisms within the host organism, without causing undue harm to the host itself. A key assertion is the possibility of synthesizing substances exhibiting a high degree of parasitotropism, that is, an affinity for parasitic organisms. This selectivity, they hypothesize, could be achieved through a meticulous understanding of the biochemical differences between the host and the parasite, allowing for the creation of magic bullets that seek out and neutralize the pathogen. They acknowledge prior attempts at chemical intervention in infectious diseases, but posit that these efforts lacked a systematic, scientifically grounded approach. This section, therefore, puts forward a bold claim that rational drug design, predicated on a deep understanding of biological mechanisms, offers a pathway to the effective treatment of infectious diseases. The strength of this claim lies in its departure from empirical approaches and its embrace of a more scientific methodology. Section 2, Preparation and Properties of Ataxyl Compounds. The second section details Ehrlich and Hata's initial investigations into arsenical compounds, specifically focusing on Ataxyl. They meticulously describe the methods employed for preparing various derivatives of Ataxyl, noting their chemical properties and their effects on spirilla in vitro. A significant assertion here is that the toxicity of Ataxyl itself could be modified through chemical manipulation. They acknowledge that Ataxyl exhibits a degree of therapeutic effect, but also emphasize its limitations, particularly its propensity to cause optic nerve damage. The authors carefully document the doses administered, observations on toxicity, and the impact on the spirilla. This section highlights a crucial early step in their research: the systematic synthesis and evaluation of a large number of compounds, seeking to identify those with improved therapeutic indices. The claim that chemical modification can alter toxicity and efficacy is supported by their detailed experimental findings, although the limitations of Ataxyl are openly acknowledged. The systematic procedure of screening different chemical formulations for varying impacts on the pathogen forms the backbone of their methodical approach. Section 3, the evaluation of arsenophenylglycine and related compounds. Ehrlich and Hata then present their findings on arsenophenylglycine and its related compounds. They claim that arsenophenylglycine demonstrates enhanced trypanocidal activity compared to Ataxyl. They meticulously document the synthetic pathways for producing these new compounds, and then describe their in vivo testing on experimentally infected animals. The importance of using standardized animal models is emphasized, allowing for a consistent and reproducible assessment of drug efficacy. The section highlights the observation that these arsenophenylglycine derivatives are more effective than Ataxyl in clearing trypanosomes from the blood. This section builds upon the prior one, presenting a concrete example of chemical modification leading to improved therapeutic outcome. The detailed protocols for animal experimentation and the careful quantification of drug efficacy provide solid evidence supporting the claim that arsenophenylglycine derivatives offer a promising advancement in trypanosome treatment. Section 4, the synthesis and examination of deoxydehymedorsenobenzyl compound 606, Asphenamine or sal, the heart of the monograph lies description of dioxidazemidoxenobenzyl, also known as compound 606, asphenamine, or more commonly, salva. Ehrlich and Hata meticulously detail the synthetic process for producing this compound, emphasizing the importance of precise control over reaction conditions. They claim that salvarsan exhibits remarkable spiriticidal activity, capable of rapidly eliminating the causative agent of syphilis from infected animals. The authors present compelling data from experiments in rabbits infected with syphilis, demonstrating the dramatic curative effects of salvarsan. They emphasize that a single injection of salvarsan can lead to the disappearance of spirichetes from the lesions and a complete resolution of the disease. The section further asserts that salvarsan possesses a favorable therapeutic index, meaning that it is effective at relatively low doses and exhibits tolerable toxicity. This claim, arguably the most significant in the entire work, is supported by extensive experimental evidence and represents the culmination of their years of systematic research. The specificity with which salvarsan acts on the spirichetes, as demonstrated in the experiments, is a testament to the validity of Ehrlich's magic bullet concept. Section 5 Toxicology and Therapeutic Index of Compound 606. Ehrlich and Hata dedicate an entire section to the toxicology of salvarsan, acknowledging the importance of carefully assessing its potential side effects. They detail the various methods employed to evaluate its toxicity, including acute and chronic toxicity studies in animals. The authors report observations on the effects of salvarsan on different organs and tissues, noting any signs of damage. They acknowledge that salvarsan can cause some adverse effects, such as fever and local reactions at the injection site, but they maintain that these effects are generally mild and transient. A key claim in this section is the determination of the therapeutic index of salvarsan, defined as the ratio of the toxic dose to the therapeutic dose. They argue that salvarsan possesses a sufficiently wide therapeutic index to make it a safe and effective drug for the treatment of syphilis. This section, while showcasing the drug's effectiveness, also demonstrates scientific rigor in addressing potential harms and balancing them against the benefit. Section 6 Clinical Trials and Initial Results in Syphilis Patients. The monograph then transitions to a discussion of the initial clinical trials of salvarsan in human patients suffering from syphilis. Ehrlich and Hata acknowledge the ethical considerations involved in these trials and emphasize the importance of obtaining informed consent. They present data from a limited number of patients, describing the clinical response to salvarsan treatment. The authors report that salvarsan treatment led to a rapid improvement in the symptoms of syphilis, including the healing of lesions and the disappearance of spirichetes. They further report that no serious adverse effects were observed in these initial trials. A significant claim in this section is the assertion that salvarsan represents a significant advance in the treatment of syphilis, offering a potentially curative therapy for a previously incurable disease. This section, while preliminary in nature, provides earlier evidence supporting the efficacy of salvarsan in humans. Section 7 Modes of Administration and Dosage Regimens. Ehrlich and Hata carefully address the optimal methods of administering salvarsan and the recommended dosage regimens. They discuss various routes of administration, including intravenous and intramuscular injection, and outline the advantages and disadvantages of each. The authors emphasize the importance of administering salvarsan in a sterile and properly diluted solution to minimize the risk of adverse reactions. They suggest precise dosages based on the patient's weight and the severity of the infection. This section is crucial in providing practical guidance for physicians seeking to utilize salvarsan in the treatment of syphilis. They argue that adherence to their recommended protocols is essential for maximizing efficacy and minimizing potential complications. This chapter underlines the importance of the technique for delivery and how critical it is for the drug's success. Section 8 Resistant Strains and the Challenge of Relapse. Ehrlich and Hata, with commendable foresight, consider the potential for the development of drug resistance. They acknowledge the possibility that spirit sheets may evolve resistance to salvarsan over time, potentially leading to treatment failures and relapse. They propose that combination therapies, involving the use of multiple drugs with different mechanisms of action, may be necessary to overcome this challenge. This section underscores the complex realities of infectious disease treatment and the need for ongoing research to address the ever-evolving nature of pathogens. The anticipation of drug resistance demonstrates a deep understanding of evolutionary biology. Section 9. The Ehrlich Hata hypothesis revisited. Toward the end of experimental chemotherapy of spirilloses, Ehrlich and Hata circle around to revisit the core claims that underscored the study. They once again emphasize the specificity of salvarsan acting upon the pathogens. While having relatively low impact on the host, they highlight the importance of creating a drug with a strong parasitotropic effect and discuss the successes that have been achieved due to that specific design. The authors also acknowledge the difficulty in creating a magic bullet that has no effect on humans whatsoever, and that trade-offs must be made. This segment is a return to the foundations of the study and the ideals that underpin Section 10. Public health implications and eradication of syphilis. Ehrlich and Hata conclude their monograph by discussing the profound public health implications of their discovery. They express optimism that salvarsan will play a crucial role in eradicating syphilis, a disease that had plagued humanity for centuries. They emphasize the importance of widespread screening and treatment programs to identify and treat infected individuals. The authors further advocate for public education campaigns to raise awareness about syphilis and to encourage people to seek medical care. This concluding section showcases the societal impact of their scientific achievement. It is a call to action, urging governments and healthcare professionals to embrace salvarsan as a powerful tool in the fight against syphilis. The authors also understood the importance of wider cultural changes that would make it easier to have open discussions about ST. Conclusion. The experimental chemotherapy of spirilloses represents a landmark achievement in the history of medicine. Ehrlich and Hata's meticulous research, culminating in the discovery of salvarsan, ushered in a new era of chemotherapy transforming the treatment of infectious diseases. Their work exemplifies the power of rational drug design, predicated on a deep understanding of biological mechanisms. While salvarsan was not without its limitations, its impact on the treatment of syphilis was undeniable. Ehrlich and Hata were not only scientists, but also visionary thinkers who foresaw the potential of chemotherapy to revolutionize medicine. Their legacy continues to inspire researchers to develop new and innovative therapies for a wide range of diseases. The systematic approach, starting from theoretical framework, synthesizing compounds, testing in animal models, and then transitioning to clinical trials, laid the groundwork for modern drug development. The work's significance lies not only in the discovery of salvarsan, but also in the establishment of a scientific methodology that continues to be followed in the pursuit of new medicines. The meticulous documentation, the critical evaluation of both efficacy and toxicity, and the thoughtful consideration of potential challenges such as drug resistance, all contribute to the enduring value of this seminal work. While future scientific developments have rendered salvarsan obsolete, the profound impact experimental chemotherapy of spirilloses exerted upon the history of medicine remains secure. It stands as a testament to the power of scientific inquiry, offering hope to those suffering from infectious diseases and setting new standards for medical research. The irony, perhaps, is that in pursuing a magic bullet, Ehrlich and Hata revealed that true progress requires not just scientific ingenuity, but also a deep understanding of the complex interplay between host, pathogen, and the environment in which they exist.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.